Anyone answer this?
Oh, yes, sir. Yes, sir.
Yes. Yes. Okay. Okay. So, okay. So we'll start with the class. So far, we have been discussing various tools used in recombinant DNA technology under biotech. So under biotech, the major topic we have is rDNA technology, recombinant DNA. How to insert the gene from other cell, one cell to another cell to produce a desired organism. For example, insulin, keep in mind, example is insulin. Human gene is inserted into bacteria, E. coli bacteria, and bacteria produce insulin. That is called the human insulin is produced. It's a very successful product. So how this involved? So we have seen few technical aspects. Few components and machines are used. This one is gel electrophoresis we are seeing, and the southern blotting technique also. Yesterday we are seeing southern blotting. What is the southern blotting? Is the name of the scientist. It's a blotting technique, transferring the DNA band into the membranous nitrocellulose sheet. So for further analysis or taking X-ray or other use, because within the gel we cannot use for the many applications. So that is why we have to transfer the DNA band into um, nitrocellulose membrane. That is southern blotting. That's one of the techniques used. One more techniques you have to know. So that is called as a PCR. It's often a repeated question. PCR technique. So first you see the PCR techniques, the video. What is PCR? PCR means polymerase chain reaction. So this is one of the main component of the DNA technology. And after that, lastly, one more things you have to know about the bioreactor. Bioreactor means it is a instrument to grow the bacteria, microbes. That's it. Bioreactor, fermenter. Okay. Then the PCR is a major techniques employed in this. So it's all in all biotechnology purpose. Even for DNA generating various purpose it is used. You know that coronavirus also can be detected using this technique. This is the confirmative test finally we will go for it. It is a RT-PCR. You remember that RT-PCR? RT means reverse transcription. Because this virus is coronavirus is a retrovirus RNA virus. But then first the RNA to be converted into DNA and the DNA to be amplified. The PCR means polymerase chain reaction. Polymerase chain reaction. So polymerization of DNA. DNA will take many copies of DNA. So many copies of DNA is taken. Then uncountable number, billions of copies can be taken, and that can be used for a wide range of applications or various diagnostic tests. So one sample, one cell is enough. So sometimes you see the crime scene, just only one cell is left, one hair particles into one cell is left. That's very valuable. So then that cell can be multiplied. It has to be the DNA has to be taken number of copies first. So it has to be taken into number of copies. Then you can use for the different testing purposes. If one test, one cell is there, if you are doing one test, simple basic test itself, that DNA is lost and very difficult to get back again. So it is application method, amplification. That means multiplication of the DNA, polymerase chain reaction. Same like a photocopy machine. The photocopy machine, what you do, the photocopy machines, we have to give the raw material, the paper we have to give the toner or the ink powder. You can join in the photocopy machines, if you give the raw material, main one is that what to be photocopied. When you are taking the initial one copy only, then you fill up with the other things like the toner, ink, paper, and other parts of paper thing. So it takes a number of copies. Same way, the DNA, first one DNA is needed. The DNA piece is inserted. It's kept there. To take a number of copies, DNA is made up of nucleotides, deoxyribonucleotides. Many deoxyribonucleotides should be given. To join this, we need a polymerase enzyme. That's called as a DNA polymerase enzyme. DNA polymerase enzyme, you learned in the this molecular biology, molecular biology, chapter 6. What is the polymerization, all the process. So, for making number of copies of DNA, we need nucleotides. There, polymerase enzyme, primer is needed. This all to be added together and the buffer solution to stabilize the whole process to maintain the particular pH or not like this we need a buffer solution is needed. So this all give input into the machine. Automatically this machine will make billions of copies within an hour. So billions of copies can be made. That is known as a PCR. This is the one which is used in the corona testing. RT-PCR, which is given, test RT means reverse. First, because DNA we are not getting from the virus directly. The virus RNA has to be converted into DNA, and then DNA has to be again multiplied and for the various testing purpose. So it's very expensive test, around approximately 4,500 rupees it takes for each PCR test. Okay, so now let us see the video. Can you see the screen now? Can you see the screen? Anyone? Anyone?
Yes, sir.
Yes, sir.
Yes, oh, thank you.
Sir, I can't hear the sound, sir.
Okay.
Sir, sir, we can't hear the audio, sir.
Okay, wait. Okay, just wait. Working on it.
Sir, you're not audible, sir.
Okay. Okay. So somewhere the problem it is here. It's okay. Volume. I can hear the volume. Full volume. I get. Then I have to read it. Just check now. Can you hear, sir?
No, sir.
Can you hear now? No. Okay. Okay. So we'll play at last. We'll check it. No, sorry. Can you can you hear now? Can you hear the voice now? Voice from the extra mark. Can you hear?
No, sir.
Anyone? Please answer.
No, sir.
No, sir.
Uh, extra mark. And redo. Just wait. Okay, now can you hear? Can you hear the voice from extra mark?
No, sir.
From extra mark, no voice. Okay, so just wait, not playing. Now it is playing.
No, sir.
No audio. So here everything is okay. Then I have to the one.
Method involves direct gene transfer.
Okay, just wait 2 minutes, please. Okay, so far we discussed the two main the instruments is electrophoresis, gel electrophoresis, southern blotting, DNA fingerprinting also we have seen the video, and lastly polymerase chain reaction. So that is an important instrument. Then after bioreactor. Bioreactor is not a big thing, it is just to hold the microbes for the growth. So you need to have nitty mediums and keep the microbes and supply the air and other necessary things. That's it. So, and put it this way. Okay, can you hear now? Okay, can you hear the voice from extra mark?
No, sir.
No, sir.
Still no. Okay, some problem is there. Please, okay, leave it till next class. I will show the video. And listen, you can take notes. Actually, if you want to from hereafter, what you can do, you can take notes and you can go slowly. This content is very good. You can also take notes. Or if you have the access expert or some art, what you can do, you can make a note later also. I think so. You have the login access. Everybody having access na extra or not? Correct? If you have access, you can go through. Okay. Otherwise, you can take a short notes on this. So far we discussed electrophoresis, southern blotting, and other methods. Now gene transfer. So gene transfer is the last part of the tools. First tool is enzymes, REN, REN or restriction endonuclease. More on REN, different types of REN restriction endonuclease like EcoR1, BAN, or so many types and then restriction site. That is the enzymes. Apart from that, other enzyme also there like ASF and alkaline phosphatase. Some other enzymes also there. Then next is the vector. We discuss on the vector. That's a big question. Last time also I asked the vector. PBR322 is the main vector, the diagram, artificial vector that you need to know the diagram, various label, various parts of the vector. So where the gene is inserted, it is normally inserted in the antibiotic resistant genes, so ORI function, ROG function. So these are the very important aspect. So second, the tool is the plasmid. Plasmid is nothing but a vector. So plasmid and vector is here is the same. Don't confuse, no? The plasmid is a vector, so that can be transferred from one organism to other. So that transfer the gene from one organism to other. That is why it is known as a vector. So a third aspect of the tool of recombinant technology is a host, competent host, and so that part we are discussing. Already we discussed in the previous slide different types of gene transfer. Already we discussed one of the class. So anyhow, we can go some more details in what is given in the external planet. So first is gene transfer. Insert the gene from one organism to another. And successful gene transfer means the recipient cell should express the inserted gene. So gene is after getting into the cell that should be able to express the gene. So inserting gene is not a big thing, but the gene has to be functional. The inserted gene has to be functional. The expressed means so it can express, it can produce a protein. For example, insulin gene is inserted. The insulin gene able to produce the insulin. That is a success. Okay, that is known as a successful transfer, gene transfer. Okay, so that we will discuss what is the various method of gene transfer. So one organism to other organism, we can transfer the gene, like a human gene to E. coli. So we use different method of gene transfer. One is normally what we discussed so far is a plasmid. Through the plasmid, we are transferring the gene from human insulin gene to the bacteria. So you take out the plasmid and take out the gene, insulin gene separately, and these two are now first cleaved. That means the place is cut, the required place is cut. That is by the then restriction intermediates and pasted with the help of enzyme called the DNA ligase. So that is done. That is now known as the rDNA plasmid. So that plasmid is now transferred through the plasmid vector as a plasmid. Other one is a different way of inserting the gene. We will be discussing. Some are direct methods, some are indirect methods. Some use vectors. So vectors will be the tumor causing bacteria can be used, retrovirus can be used. So different agents can be used to transfer the gene. So these are indirect method of gene transfer. And sometimes directly we can insert the gene into the organism. That is called the direct method. The direct method means using some chemicals. Like the last calcium chloride or divalent cation or simply calcium is used. Calcium and heat shock, 42-degree ice, you have to bring from the ice, then 42 degree for 2 minutes, again back to ice, then 42 degree, then back to ice. So if you give this is known as a heat shock. Along with the calcium, it forms a pore, it increases the pore size, so plastic will enter. So directly enter, because we are giving external from condition heat shock, we are not using any vector. So this is a direct method. Or electric voltage, high voltage we are giving, high voltage, that high voltage power amplitude, and then that increase the pore size. So instead of calcium and then heat, the voltage can also it work. High voltage if you give for short duration, that increase the pore size, and then the plasmid can go inside. This is called the electroporation. Electroporation. Giving electric current and the pores are formed. So temporary pores are formed again in the data. So like the gene gun and other methods, micro injections. These are the methods where directly, forcibly you are entering the gene, inserting the gene into the host cell. So these are the different method of gene transfer, direct and indirect methods. So then if you want, you can take a notes also here. So these are class. Okay, now, so direct method and performed directly, but indirect methods are performed through the vectors and other kernels. So in indirect methods, we discussed the plasmid or natural gene transfer. So here, like natural virulent prokaryotic organisms like bacteria, tumor-causing bacteria, we discussed earlier, agrobacterium tumefaciens, TA plasmid, tumor-producing plasmids. So these are the virulent bacteria, very causing bacteria, which can transfer the gene into the host cells, like tumor-causing bacteria. What we can do in that bacteria, original genome is taken, and then the tumor causing gene is removed. In place of that, we keep a gene of interest. What gene we want to keep it. Then this bacteria is allowed to infect the cell. It infects the cell, that is called the first infected cell, it's insert the gene, that is insertion. After insertion, it causes infection in the host cell. But instead of causing tumor, but actually here it insert the gene of interest. So then the cell, host cell is now transformed. Transform means the host cell can incorporate the gene inserted. So this way gene transfer can be done to other agents. These are the indirect method using vectors, viruses, bacteria. These are the indirect. So you can see the disease causing region of the genome, the DNA. So suppose particularly the disease causing, it causes a tumor, it's a virus, retrovirus, RNA virus. So what happens, that particular part is removed, that part tumor causing gene is removed, it's not harmful now. In that case, we keep the genome, what gene want to insert it. So this also through virus, through bacteria, or through plasmid. So these are the methods known as the indirect method. Here all they use vectors or other reagents. Okay, so in this method, this direct method, there is no vector. Over here, no vector is used. So the directly gene of interest is introduced into the host cells. And various methods, electroporation, chemical method, microinjection, gene gun. Some methods already we discussed. So electroporation is giving high voltage. When you give the high voltage, the pores form, then it takes up. Chemical method means they use different chemicals, like the calcium chloride or other heat shock. Heat method also there one more you can add it. And the polyethylene glycol commonly used for this purpose, insert the gene, that is used for chemical method. Micro injection, so last time we that is right now, so inject that very fine, inject the DNA into the mostly in the animal cell. So normally this is animal inject. Gene gun, so the bullet, I said no, the micro bullet. So that is used 0.1 micron, that I enter, and that can be used at the speed of 1400 feet per second. The particular speed they shot, and then because the diamond and the tungsten, these are the inert metals. Inert metals, these are coated with the DNA. So along with that, the metal, because we need something, to go take up the DNA, the solid substance. They take inert substance like tungsten, and the whole structure, which is coated with the DNA, then it is inserted into the nucleus. Right? That is called the gene gun method. These methods are directly inserting, no vector. What is the first point here? There is no vector is involved. So these are the direct method. Sometimes the question the need or we have to identify. So which is a direct or which is indirect methods. So electroporation, chemical method, microinjection, or gene gun. So these all the direct method of the gene transfer. So electroporation, name itself is that electron use electricity. Electrical impulses are used in high field strength, and impulses are used some electric power is used, current is used, high voltage type. So impulses are used, and that causes poration. Poration means forming the pores or holes in the host for equally. So normally you will not take up. So the reason is there that the cell membrane is there will not allow the DNA. So to take up inside. You need from a small holes. So the hole is into the electrical field that is called as the electroporation method. Okay, here chemical method, the PEG, the abbreviation is polyethylene glycol, the PEG is used. This may come to the exam. So this is the chemical method. They use some medium, some carrier to carry the gene. These are the chemicals carrier. This is called chemical method. These are the inert, it's not causing any changes in the organism, like tungsten, gold, these are inert substance. So, and this can carry the gene, introduce the gene into the whole cell. So chemical method is PEG. So the micropipette means it is a needle, very fine needles. You see the thickness, 0.05 micron. That is thin, very thin hair-like needle, very minute needle. It's inserted. Normally, it is inserted in animal cells like egg. Directly, the gene is placed with the help of injection. So here what happened, DNA is coated with the inert metals. I had to wait for the voice to complete this because external voice today we could not hear because I had to restart, shut down the system and restart my work. The video started at the time we can take 5-10 minutes yesterday. So that's why I started. Maybe next class I will be ready. Today you can listen or you can take a notes also. Notes is very important. You can take a notebook or a pen, you can just key points you can note it down. That is very important. So gene gun or biologistic method, here mainly the tungsten and gold is used. Sometimes it comes to the exam like what is used, tungsten and gold. These are inert metals are used, and these metals are coated with the DNA. So it is like that. Suppose you want to pass one paper to What we do is for example, next place, next house, what happened, you cannot throw the paper. What you try with the stone or something, okay, then it goes up. So like this way, we need the carrier. The DNA is very light, no? Need a carrier, that is metal is used, inert metal is used, that will not cause any harm here. So then along with it, it has to be inserted. For inserting gene, then what particular speed has to be maintained. So it can be applicable only for the plants. Remember, this can be applicable for the plants only. And the particular speed, 1,400 feet per second. So that much speed velocity, we have to charge this the coded tungsten bullet coded with the DNA. It can directly go to the nucleus. So the DNA again will incorporate with the genome of the host cell. So this is the method is known as the gene gun or parallelistic method. So this method you can see it is a gene gun method. These are the white one here you can see the gray color. These are the gold tungsten bullets, and the green ones are the desired DNA. The DNA to be inserted. The green ones you can see it is coated with the DNA that can be inserted into this at a particular speed. If it is over speed, it will come out of the cell. Less speed, it comes back. No, it has to be correctly maintained the speed. Then it can go into the nucleus. Or this entry, this side you can find the entry by transformation. So the directly it can enter by using like heat shock or electroporations. That method also can be used. It can make a pore, and then through the pore it can enter inside and can transform the cell. That means it's called transform. After taking up, the cell is known as the transformed cell. So the green color is the gene of interest is inserted. Or micro injection. Here you can see the micro injection using a very fine needle, 0.05 micron, no? The diameter of the needle. So that can be inserted and can be injected directly into the host cell. Then it can be used by the virus, this is called vectors, virus or bacteria can be used to transfer the gene. So these are the various way of transferring the genes into the host. Some are direct method, some are indirect method. And what are the direct method, indirect method, different types you have to know. You make a point, you can know, and then so many questions are possible. Like for the tungsten, polyethylene, or and the electroporation is due to what using electric current, electric voltage, that is in electrical impulse, the short impulse vibration, so then it causes a pulse. And earlier method, the traditional conventional method is the calcium, divalent calcium is used again with the 42-degree heat shock, then bring to ice, 0 to 42, 0 42. So then like that is not a heat shock. So this way the gene can be inserted. After inserting, you can see the below this diagram. In the diagram, the gene is inserted. The green color one is the gene is inserted. That is original gene, plasmid, and then the gene is green one is the inserted gene. Now this is known as a recombinant post. Recombinant post. The DNA is recombinant DNA. So this way the gene is transferred. So after transferring gene, what is the next process? We have to select whether the bacteria is taken up the gene or not. That is using the marker. That also we discussed it. Using the marker and with the different antibiotic resistant gene, or it can be used in the inserting the gene into the actual the gene code for the protein, like a LAC-Z gene. So we discussed LAC-Z gene. That is forming the beta galactosidase enzyme that will not produce, so that will not give color, white color, so that will give the only white color, and that is known as a blue and white method, upstream method, different method. So identification of the recombinant host, which is the host which has taken up actually the recombinant DNA. Then that host, that bacteria has to be multiplied. So that is the last, before last part. So multiply using the fermenters, bioreactors. After that, the desired product is obtained, purified, and marketed. So these are the various components of the biotech. The next class we'll discuss all in sequence. Okay, can you just answer? Anybody?
B, sir.
Okay, B, very good. So why it is a direct transfer? Why it is direct transfer? That means no vector is used. That is right. No vector is used. Inserted directly. We put this in the bacteria in the solution and the plasmid also in the solution. For example, we pass the current, give a little impulse, no? Like a high voltage impulse, and short duration, very brief duration, then immediately forms a pore. So then through that we can enter. Directly it can just give to the we give something the stress factor. We give causing stress to the bacteria, it forms a pore. So like this. So like cause of irritations, then it forms a pore that immediately get there. So this way the heat shock or electric shock can be given. So the electro portion is giving electrical shock. This is a giving electrical shock and then that helps in forming the pores in the bacteria. So immediately through the pore, the plasmid is inserted, taken up inside. So this is known as electroporation. This is a directed gene transfer. Now vector is involved. That is why it is direct. And detail you can see. So what is a electroporation? Okay, today actually I wanted to complete the PCR and other techniques of the gene transfer and other, but due to some technical issues, could not, again running short of time, you cannot again shut down and restart. So we will take it some more time. So you can note down next class what you can do. You can note down if you want, you can just interrupt and tell me whether you go slowly. You can have a notebook with pen, everybody, and to make a short notes, repeat, you can make a note, that's very useful. No, I will ask you questions. I will ask hereafter, I will ask you more questions after I reach one topic, or you also can tell me, bring me. So wherever you want to me to stop, I will stop it, I will redo, and slowly we go, you can take notes also. So some of the content is very good. Even the PDF also, what I am sending, that also good, that also same NCRT note, it's nothing different. Okay, then you can use that extra mark also you can use it. You can take a note, then easy to answer. Easily you can follow the topics. And if you don't understand, you can just tell me. I will repeat. Okay, so far what we discussed, all it covered in this topic. This is the last big topic, this big question. And so far we have completed all the aspects, all the steps in assorted way. Here and there we completed all the one in different ways. For example, now, first point I discussed, but now we will know detail. Second point is that about restriction engine already we discussed fully. Okay, and the third point is insertion of DNA into suitable vector forming recombinant. That also we discussed it. Cut with the restriction enzyme, forming sticky end. Same the insulin gene also cut with the same enzyme. There also it forms a sticky end and pasting with the ligase. So this part also already done. And the fourth part, transformation of host cell. Transfer into host cell. So just now also we discussed, gene transfer, different method of transferring the plasmid into the host cell. So like, but here we are studying all in sequence. What are the steps in recombinant technology? In between one place, we have the major topic is the polymerase chain reaction. That will be the big question. Since the audio is not clear, not working, next class we will discuss about the PCR. So we see the what is the other steps. So these are the complete steps in followed during DNA recombinant DNA technology. Okay, so now what is the fifth one? Identification of the transformer host cell from culture. So with the help of marker, that also we discussed in detail. One is using antibiotic resistant gene. In the PBR 322, what is there? The two genes, two antibiotic resistant genes, remember ampicillin, tetracycline genes are there. You can place it anywhere. Then there it needs two types of antibiotic. We have to make it two places. One is tetracycline, other is ampicillin. So then we need to segregate the transformant or non-transformant. Or the one direct method is there, is using lac reset gene is inserted, it's splitted and the gene is inserted. It's called the insertional inactivation. So then the gene is not lac reset gene is not produced. If it is transformant, the lac reset gene will not produce the beta galactosidase. So the white color is appeared. That means that it will not respond to the chromogenic substance. And another one is non-transformant, will produce a beta galactosidase that will get with the chromogenic substance, gives a blue color. Blue color colony is non-transformant, white color colony is a transformant. So these are the different way of identifying the transformed host cell. So after that, when this transformed host cell you are selected, for example, like in the blue-white method, blue eliminate all the blue colony, take up the white colonies for the other cell. Across it forms a small colonies, catch one. So like dark forms. Select the white colonies. These are the transformant, and then directly we can go to the step 7, multiplication of the host cell. This is using bioreactor, big vessels, fermenters. So there it is produced, and so they have suitable like inducers are used. Insulin means they have to use some glucose. Then that will use production of insulin. Like you know that now in the beta galactosidase, when it is produced, when lactose is given, lactose acts as a inducer, the lactocron. So then like that inducer is given, then we produce the insulin. The insulin last step is there, the obtain the gene product. So then the gene, the insulin or product is segregated, filtered, and then processed, purified, and then packed and sent to the market. So these are the things. And in between, step number 6 is amplification of recombinant DNA. So the DNA, once if we get the desired gene, only one gene is one cell will only be obtained. For example, remember the kind gene, only rarely required one cell. So that's not the spoiled. That can be used for one test, then so we cannot get back again. So that before doing many tests, many things, many analysis, what happened, we have to take enough copy of this. To take enough copy from the single DNA, a single fragment, to take a billions, uncountable number of copy, that is PCR is used, amplification of recombinant DNA. So that is a new in this. Remaining all already we discussed, except the first point and the last, the 6, 7, 8. We are going to discuss. In between already we discussed. And okay, so from this topic we will discuss tomorrow. Already it's getting late, but just 2 minutes. 2 minutes, I can explain what is this isolation of purification. Then I will repeat during the next class. Isolation and purification of DNA. So now we want to do the RDNA technology. What happened? Not nowadays directly we get the gene. We get the gene, insulin gene. That is a different aspect. But you should know the procedure. The insulin gene is not available as per the Human Genome Project. It is there actually. But what we take off from the human cell and then prepare the insulin gene and insert it into the plasmid and then multiply. The complete process how we have to do. First, the DNA is purified. The cell is obtained. The DNA is purified. How it is purified? By using different enzymes. The DNA from the cell. So one cell we are taking from the human cheek cells. From inner cheek cells we are taking. So select one or two cells. What happened? That cell has to be added with a different enzyme. Different enzyme to be added to digest other parts, cell membrane, other all cell organs to be dissolved. Leaving only DNA. We should not put DNase. DNase is the enzyme which digests the DNA. You can put RNAs, RNA can be digested. You put protease, protein can be digested, like histone can be digested. You put lipase, cell membrane will be digested. So like this. So various enzymes according to the cell, plant cell, animal cell. So plant cell means additionally they have cell wall. You need to add cellulase. Okay, fungus cell means pectinase enzyme to be added. Remember that is known as the isolation of a DNA. So from the cell, we have to separate the DNA alone. The easy method is simply you have to add enzymes. Don't add DNAs. Okay, so like this, so cell wall can be digested. If it is a, you are taking DNA from the bacteria, you have to do the lysozyme first. And histone can be removed. Histone is a protein, it can be removed by protease enzyme. So this way, so this we will be discussing. Finally, we get only the DNA. So DNA is left, the DNA can be removed. You can see here the DNA. Suppose you are taking the DNA from the bacteria, what to remove the cell walls or the capsule, what you have to use? Lysozyme can be used. If it is animal cell, it is a other directly, like this protease can be used, or it can be plant cell, is a cellulase can be used. So like that. Then the DNA will be separated. Still the histone is there. The histones, because they form the nucleosome, remember the histones are positive and DNA is negative charge, they form the coil around it. The histones are removed with the help of a protein digesting enzymes. Treatment with the proteases. See, one of the wherever the RNA is there, RNA oligonuclease. RNAs can be used for like this RNA. So only the DNA is left. You can see here the DNA thread. So this way you will get. So this is your practical actually. Instead of this, all the enzymes, what you can do, soap solution. Simply easily available solution is a liquid wash. So take a liquid wash, soap solution, and then you add acetone from the test tube. That's it. Then you will check it. They all will be dissolved. The protein and the lipids all will be dissolved and leaving the DNA. So DNA usually from mixing the ethanol, you will get the precipitate of DNA. So it's like this called as a schooling technique. So this we can take a DNA thread. This is one of the practical in class 12. So we will start this practical after we finish the class. You can see the thread-like structures, white thread formed, and then that can be taken out with the stick. So this is the DNA thread. Now, okay, time is really. So we'll be discussing this in the next class. Anything? Any doubt? Any doubt? Anything to discuss? So today could not play because of the audio problem. Maybe I will make sure next class will be yes, yes. Anybody want to say anything? Hello. Okay, next class you can take a notebook and the pen and you can take a note. You can go slowly also. If you don't understand any concept, you can tell me. I'll stop it and I will do it. Okay, thank you. Thank you for meeting.
Thank you, sir. Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
Yes.
Thank you, sir.